So good morning, or good afternoon, or even good night, depending on your time zone. I'm José Abreu. I'm part of the APECS. I'm a PhD student, and I will be one of the chairs with my friend and colleague Guilherme.
Hi everyone, good morning. Hope you guys are excited for this. So my name is Guilherme. I'm a PhD student in Aveiro University, Portugal. And that's it. Enjoy everyone. Thank you.
So to start this workshop session, we have with us João Carvalho. João is a biologist and a researcher at the University of Aveiro, where he started his academic path and continued throughout his PhD. During this time, he has collaborated with the University of Barcelona as well with the Zoological Society of London. Since then, he has taught at the University of Florio in Mozambique and in the University of Porto in Portugal. Currently, he does his research and teach at the University of Aveiro in Portugal. João is also the editorial coordinator of Lucanos, a journal that brings relevant environmental and social themes together. His research focuses mainly at the population and ecosystems dynamics, resource management and conservation. In addition, João has a strong background in statistics and geographic information systems, as maps, GPS, and that kind of data. So therefore, in name of APECS Portugal, we thank João for being here. And we hope that these sessions finds you all well and contributes to improve your skills. So please note that this session is being recorded. Also, in the end, we will ask to take a photo group, if you don't mind. Finally, as my colleague Hugo said, we're going to ask to all the participants to have the micro and the camera off during the session. Whenever you want to raise a question, you can raise your hand, that Zoom option, or you can write in the chat that we will put directly the question to João. So I think that is all. Enjoy and have fun, and thanks for participating, and thank you. I will pass now to João.
Okay, José, thank you very much. Dear colleagues, I hope you are well and sound, of course. First of all, my English is far from being perfect. So if you have some doubts, if I'm not totally clear, feel free to interrupt me whenever you want, okay? I will be there, I will be here, and I ask your questions now or at the end of the session, okay? So no problem with that. Feel free to ask me. Well, the presentation, José made my presentation, is okay. I'm a junior researcher from the Aveiro. I finished my PhD, well, 2 years ago. Yeah, time, okay. But okay, I'm a junior researcher, and I would like to thank the invitation to be here. Congratulate APECS for this amazing initiative, and particularly to Guilherme Jeremias and José, because I have contact with them, and for his professionalism, commitment, and the organization. It was everything nice and amazing. And thank you. Well, to start, I'm not an artist, and I'm just trying to be a scientist. I'm a junior researcher. And therefore, one may ask, well, if you are not an artist, what are you doing here in a workshop entitled Art and Science, Your Research Through Image? And this is a good question, but maybe over the next hour, more or less, I don't know how many time I need, but for sure less than 1 hour and a half, I will try to show you how search engines, software, maps, graphs, networks too, could help you in synthesizing and communicating scientific information, okay? I obviously hope you find it useful. And now we are in the era of information overload. For instance, now we realize COVID-19, for instance, if you made a search in Google, Web of Science, Scopus, we feel overwhelmed by the amount of information that we are facing. And what is right, what is meaningful, how could we sum up all the evidences? Well, I'm trying to show you some tools and some softwares that could help you in this endeavor, okay, in this challenge. Because synthesis, evidence synthesis, is right. This involves combining ideas or results from 2 or more sources of information in a meaningful way. So we have 2 or more sources. In fact, we can have thousands of sources, thousands of articles, national reports, European reports, official reports, and evidence synthesis is the process of bringing together information and knowledge in a meaningful way to support decisions and policies, okay? For artists, it could be done in a beautiful way. For guys like me, you have to use software, you have to use some, and if you work together with artists, well, so promoting synergies, we can do great things together, okay? But first, before I start the tutorial, start to talk about synthesis, I would like to take advantage of being the first speaker to present, if you don't mind, of course, a brief history of the art and science through time. 10, 15 minutes, maybe 20, okay? Because now art and science and humanities, I guess it's a question of time, but do not seem to have many points of contact. And this is why I found this initiative excellent and timely. Okay, the school of thought in scientific areas became mainly driven by specialization, hypothesis, and data, and no problem with that because science is hypothesis driven, it's data driven, must be. However, we feel overwhelmed, as I stated before, by information. We feel under pressure to publish articles. We feel under pressure to gather citations, to be recognized and respected by our peers. And with this pressure, okay, the wonder about science, okay, who feels wonder about science? It's difficult. And art by itself follow its course, a different course. And therefore art and science may seem a bit more distant than ever now. And this is why this initiative is important. And maybe we should come back to the essence of art and science to see how close they are, okay? Because art and science and both disciplines share the interpretation and representation of nature. And now I will share my screen, okay? Can you see it?
Yes, we can.
Yes. So nice. Okay. Are you seeing the presentation? Yes?
Yes.
Okay, perfect. And this is, okay, so let's go back to the essence of art and science. And maybe, I don't know where art and science, when art and science become, or art and nature become a matter of interest for men and for societies, but for sure, for instance, back into 25,000 years ago, to the Paleolithic period, we can see these engravings. These engravings dates back from the Paleolithic period, and they're a nice representation of nature, of animals from which humans and men at that time, they are dependent. And these engravings can be found here in Portugal, in Coa Valley, near the boundary with Spain at center north of Portugal. And it's part of a complex of engravings and paintings that include also Altamira in Spain, Lascaux, France. And it's nice, and it's really, really amazing how with rocks, and they could represent horses, ibexes, deers, and all the animals they depend at that time. And this may be the first representation and first expression of art at that time. And if you pay attention at the image at your right here, you see that this image also represents a kind of dynamics, a kind of motion. You have a horse here, the body, the neck, the muscle. The first image, we can see the horse grazing, then raise the head, and that third image, third head here is patrolling, is looking around. So this sense of motion it's already present, and sense of art was already present in the Paleolithic period. So, and art, science, societies have evolved through time. Sometimes with promising periods, sometimes with not so promising periods. And dark ages were perhaps one of the ages where this merge of art and science were not so promising. But after dark ages, we found the Renaissance and the Age of Enlightenment, that are some of the periods where the nature of art and science were best explored. The aptitude for wonder were higher than ever. For instance, we can remember Michelangelo, Donatello, Leonardo da Vinci, just a few examples of the amazing scientists, artists, explorers, creators that emerged at that time. But not only. And now I will present some chronological examples of art and science through time. But these examples also represent a way of synthesize evidences, a way of synthesize knowledge in a beautiful and artistic way, okay? And for instance, this guy, Andreas Vesalius, was born in Brussels, I guess, Netherlands, and represented in this book a great and challenging images about human anatomy. And at that time, this book was a reference book, and he calls it entitled "The Humani Corporis Fabrica." And this kind of images, this kind of representations, we need more now. Okay, images to cross boundaries, to be talked about, to be mimicked, to be inspired. Okay, because doing so, it will result such a cultural effect that could be that will be talked through times. And this kind of example is focused on human anatomy, but we have a lot of examples from human anatomy, from nature, from solar system, and you'll see some other right now, because there are many examples of art and science intermingled based on observation and interpretation. Okay, for instance, this example from a Portuguese cartographer, Bartolomeu Velho. Okay, apart from it being wrong, it looks perfect. Who can say that this is wrong? It's beautiful, okay? Anyone may feel impelled to believe in the Ptolemaic center, in the Ptolemaic theory of the universe, okay? So Bartolomeu Velho was a 16th century cartographer and represents here the solar system, well, according to Ptolemaic geocentric system, with the Earth at the center of the universe. But art is represented here better than ever. For instance, other example using maps. This map was developed and was okay. This map is from João Blow. It's my background image. João Blow was a Dutch cartographer, and this world map is entitled Nova e Accuratissima Terrarum Orbis Tabulae, and incorporates the discoveries of Abel Tasman and depicts for the first time the solar system according to the heliocentric system of Copernicus. Okay, it's the first map that incorporates this representation. Here we can see an heliocentric theory here represented in this map, okay, which show the Earth revolving around the Sun, okay. And although Copernicus' groundbreaking book on the revolution of spheres had been first printed in 1543, just over a century earlier, Blow represented this kind of information, these new evidences in this amazing representation. Okay, but now from the Earth, okay, we all feel wonder about the variety of forms, colors, shapes that Earth has to offer to us. And here we have a great example of a naturalist, of a researcher too, of Maria Sibylla Merian, born in the 17th century. And for Merian, observation, science, and art were all part of the same process, okay? And their paintings were both a way to communicate and understand the processes of metamorphosis. For instance, this image at your left, and you have these images that at that time was very criticized because show a spider hitting a hummingbird. That time was impossible, but later was confirmed. And this is what amazing example of how hard science could be 4 centuries ago. So it's amazing, okay? Backing to the solar system and to the universe once again, and art and science can allow to move between dimensions very easy. We can, because it's in a chronological order, and can you see that maps and graphs are really useful over the history apart from art. Thomas Wright was an English astronomer, mathematician, and Wright published an original theory or new hypothesis of the universe in 1750 and explained the Milky Way, for instance, and represents it in 2 and 3 dimensions. And this guy, Thomas Wright, was frequently mentioned in the books of Carl Sagan. In the recent book of Anne Druyan's, the wife of Sagan, about memories of Carl Sagan, Thomas Wright is named often, because he represented the universe and solar system in the 2 and 3 dimensions, and motions and moons, usually according to the heliocentric theory. So representing this information, not in a draw, not in 2 dimensions, but in 3 dimensions, it's amazing. And Thomas Wright did that at middle of 18th century. So this is just another example of how art could be used. And this is perhaps the father of biogeography. We have to have a father for everything, and maybe Alexander von Humboldt is the father of biogeography. I don't know if you have already read The Invasion of Nature from Andrea Wolff. This is an amazing biography of Humboldt journeys and achievements. This is a great book. And for guys that love to be seated in a couch while traveling and reading, this is the right book to do that, because it's really important and amazing book that show the importance and the influence of Humboldt for the researchers at that time and for future researchers, from Darwin and so on. Okay, because Humboldt traveled extensively in the Americas, exploring and describing them for the first time. Describing landscapes, describing animals, several species from a modern scientific point of view, and more. Humboldt was too close of different artists and writers, John von Goethe, for instance, Thomas Jefferson, the President of the United States. Was a person that combines and synergies from a lot of research fields and art, and it was very important at that time. In these illustrations, the Naturgemalt represents the Chimborazo and Cotopaxi mountain volcanoes, south of America, a cross section, where it is represented as a nation of vegetation types, species, and it's said that constitutes the foundations for the field of biogeography, this map, and it's really, really beautiful. As you can see, this is an image of 2 volcanoes, a cross section with the description of species, the zonation, and it was first published in 1987 in Geography of Plants. Okay, and as I said, it's foundations for the field of biogeography, and this is a beautiful way to synthesize evidences, to synthesize and communicate evidences. Apart from being an explorer and a naturalist, Humboldt was also a communicator. Okay, was very excited communicator, was amazing, in fact. Okay, and well, the context we born is important, of course, and at that time more than ever, because we can ask me how they financed their expeditions. Much of these expeditions were self-financed. Okay, and so the context we raise, context we born, if we belong to a family, a rich family or not, it was important at that time. But there are some nice exceptions, really nice exceptions, and a lot of exceptions. And maybe Michael Faraday, that was not a naturalist, that was not an artist, but just to say that it's one of the main exceptions, showing that even receiving little formal education and even as little funding, we can seek and we can achieve great findings. Faraday established, for instance, the foundations for James Clerk Maxwell from late classical theory of electromagnetism. So it's important to be this kind of notion and this kind of idea about science in the past. Because when I found myself alone and talking with my colleagues, we said, hey, well, be a scientist today is a challenge. And it is, but be a scientist or a naturalist 2 centuries or 3 centuries ago, it's even much more difficult. Okay, so okay, it's not difficult, but we have to take advantage of some facilities we have now to try to produce something really nice. Okay, and this is another example. Now Anna Atkins. Anna Atkins was an English botanist and photographer. This is first person, she's the first person, considered the first person to publish a book illustrated with photographic images. So it's a great achievement. Okay, it's the first book titled Photographs of British Algae, Cyanotype Impressions in October 1843. So first book with the science, the art, the technology has evolved through time, and this is another important achievement. Okay, and when we are talking about science and art, we cannot forget, of course, Ernst Haeckel, and I guess you know them. You know him very well. Ernst Haeckel was a German zoologist. He published several multicolor illustrations of animals, of sea animals, mainly collected in a book, Arts Forms of Nature. And although Haeckel was an important evolutionary biologist, his work was best recognized and is competent in invertebrate anatomists. Okay, so this is a great example of artists that combine art and science in a meaningful way in this kind. Well, to finish, now much more recently, in 1953, Watson and Crick, well, they deduced the double helix of DNA and represented in a 3D model, and this is one of the most important models in the era of biology because it marks a turning point of modern biology. So this is just another example of how, so we see that art can be applied from planet, solar system, universe, from plants, from molecules. So art and science evolved through times in different research fields, and we should take advantage of these synergies to promote better science, to promote the communication between science and society, to lead science to other publics, okay, beyond the academy. And of course, we have other examples, a lot of examples. William Shadelson was an English surgeon and teacher of anatomy, surgery. He published Osteographia, that is a nice book of bones, okay. John James Audubon was an American ornithologist, naturalist, and painter. He combines interests in ornithology and art to produce and to make a complete pictorial record of all bird species of North America. It's amazing. I love it. It's the illustrations are really, really beautiful. Martha Maxwell was an important American naturalist that constituted the foundations of taxidermy, okay, of modern taxidermy. And we know how taxidermy is important for teaching, for museums, and this is other kind of art. And of course, nuestro hermano Santiago Ramón y Cajal was an important neuroscientist, pathologist, and described with great art and great imagination the brain, the links, the connections between neurons, synapses, and everything else. If you can take a look to this book, to the book Beautiful Brain, the drawings of Santiago Ramón y Cajal, is a Nobel laureate, you will become really, really impressed. And you have others. I have here a list of many others. From Joseph Banks, was a British botanist. Humphrey Davy, really impressive. The Davy lamp is very known. Alexander Volta, Dmitry Mendeleev from the periodic law. All guys, all scientists that represented science, their findings in a synthetic way, in a synthetic and beautiful way. In Portugal, we also have Bartolomeu Guzmão, Abel Salazar, and so on. We have several examples that could be inspired, very inspired for us, okay? Well, just some recommended books to foster our wonder about science. There are too many, but This one, The Greatest Show on Earth from Richard Dawkins, is beautiful. The Invasion of Nature is really nice. The Meaning of Human Existence for Edward O. Wilson and he has some chapters addressing this importance of connecting humanities and science. We have Steven Pinker, Enlightenment Now, but New Intelligence Too, and The Age of Wonder is really amazing from Richard Holmes. Okay, so books focus in the Age of Enlightenment, the Renaissance, and the wonder of science. We feel science, and we need this now most than ever. Okay, we must feel wonder about science, because otherwise we are producing knowledge, but without wonder it's more difficult. Okay, we should feel inspired, and it's really important. But now maps. Let's talk about maps. Okay, what about maps, graphs, and networks? And this is the essence of why APECS and Guilherme invited me. Okay, to show and to talk about maps, because maps have an important role in development and representatives of knowledge. We already see it, but also on health. For instance, in the class of spatial analysis, in the classes of spatial analysis in geography, this is commonly the first example of the importance of maps and spatial analysis for human societies. And John Snow, doctor from London, a British, is viewed by many as a pioneer of disease mapping. This is a map of Soho in London, where cholera outbreak started in 1854, and no one was able to identify the origin of this outbreak. Then John Snow seated in his desk, put a map in front, and georeferenced all cases detected of cholera. And with a simple map, a simple but beautiful map, he was able to recognize the source of the infection. Okay, and there was a water pump. They closed the water pump here. Okay, they closed water pump, and the cholera outbreak was controlled. Well, this is an important example of how maps could help not only to represent science, but also to solve real world problems, or local problems. Okay, and maps, you can also see some nice books. There are a lot of nice books. One of my main favorite authors is Jerry Bruton's, and we can, if you have curiosity, A History of the World in 12 Maps is beautiful. It's a nice book, or even better, Great Maps, the World Masterpieces Explored and Explained. Is beautiful. And I have other one, A Map of the world according to illustrators and storytellers. Are amazing maps. Okay, amazing maps where the knowledge of geographers, artists are close together. Okay, so art and science go hand in hand. Well, in the world of Richard Dawkins, an appetite for wonder is needed to new and great achievements. This, and of course some blood, tears, and sweat. Okay, currently my main concern is the current system replaced wonder by publication number of metrics. Today there is so many information that the student, the researcher, should be able to sum up and represent what we need most. Okay, we are starting a master thesis, we are starting a PhD, and we feel overwhelmed. Well, I have these objectives to answer, but I have so many information that we should be able to select what you want, what could be useful for us. Okay, and I will show you some tools to do that, and this is why I bring this presentation. Okay, because to make sense of the data, we need creative approaches, and art, science can lend new representation of previously incomprehensive forms to represent in transformative ways. Okay, art, maps, graphs, we can do that in ways to transform incomprehensible forms and interpretable forms to help science, to help policies and decision making. Okay, and this is why I'm bringing this presentation, synthesizing and communicating evidences in scientific research. We already saw that during the history it was used thoroughly or widely used by different scientists, but now we are in a era of information, and therefore we should be able to know the sources of information and to perform great and useful and meaningful and applicable synthesis. Okay, so just know we are experiencing, of course, an information overload. Okay, we have a lot of information, and the collection of information and the publications of articles are underway. Currently, empiricists and synthesists don't work in close contact, and this is one of the main problems. Okay, currently we have empiricists from one side and synthesists from the other side. Okay, and data analysts and curators are needed more than ever in several scientific fields. For instance, here in Portugal recently, we experienced a controversial issue was installed because the data about COVID-19 patients were not reliable enough. We have pregnant men, we have guys that died in March but were alive in May. So it's a problem, it's a really problem, because empiricists and synthesists and data curators don't work in close contact, and therefore to produce reliable reports is difficult. Okay, so in the future, one way to overcome this issue, empiricists and synthesists must work in close contact. The data should be available even when unpublished, as data-driven science demands co-producing knowledge, because there are issues too complex, and therefore these too complex issues should be analyzed ideally for different teams to see if the results are the same or not. Okay, and of course evidence synthesis should be fast, open, and completely transparent. Okay, and this is surprising how the theory, bearing in mind that the theoretical and practical importance of data synthesis and presentation is really important. It seems surprising how that analysis didn't really get as going as a science. And part of the problem is that science and empiricists and data curators still don't work in close contact. Okay, but step by step, this is a nice article from Nakagawa published now in 2020 that states this, precisely this. Okay, we have a lot of information, but the information is not systematic collected, it's not systematic reported, and therefore it's difficult to gather conclusions about that. Okay, so to perform a synthesis, we should know what we want to answer and what you want to synthesize. Okay, we are starting a PhD thesis, and I want to work in a first chapter about the state of art of a particular issue. So we need to have a particular question, a specific question to answer. Okay, and therefore we could perform this kind of search, and to perform a transparent search, a rigorous search to develop an accurate, consistent, and unbiased synthesis. Okay, and there's a paper throughout the presentation. I leave some comments, I leave some links. I can share the presentation with you later, and you can find some tutorials, some nice articles to read and to study. And there is a nice paper published in 2018 for Chris O'Donnelly, and now I will read, because he stated a statement of a sentence that is really important about data synthesis, about data representation. And Chris O'Donnelly said, and colleagues said, an accurate, consistent, and unbiased synthesis of the available evidences is arguably one of the most valuable contributions of a research community can offer decision makers. Few understand how to produce or commission good synthesis, and too many are reaching for information that is out of date, incomplete, or biased, sometimes from just one study or researcher. Okay, now we don't have this problem. We don't have, because we have a lot of sources, we have a lot of researchers working for the same purpose. Even where good synthesis exists, they are often not available quickly enough. In the realm of public policy, it may be not that a good enough version available before a decision is made is much more valuable than a perfect version that arrives a day too late. Issues range from the impact of pesticides on pollinators to who should be kept in quarantine during the disease outbreak. So this is amazing because it was written in 2018. And according to these authors, okay, based on these authors, synthesis should involve 4 baseline principles. And this is important for you guys that are working or want to work in synthesis or work to develop or write a chapter about synthesis. And a synthesizing evidence should be based in 4 main principles. Should be inclusive, okay? For instance, you have a research question, you identify main stakeholders that can be interested in this question. You should involve with these policymakers and stakeholders to help you in producing relevant and useful outputs, okay? Inclusive, should be rigorous, okay? Use most comprehensive databases, and we have a lot of information, a lot of peer-reviewed papers, national reports, official reports, European reports. So we should use most rigorous information and available, okay? Should be clearly transparent, should be clear and transparent, okay? Should be described the research question, the methods used, and this is how important. For instance, when you use Web of Science, and the example I will give you, we'll use Web of Science, you use strings to search certain articles. This should be totally clear because other person could be replicate your search, okay? So a research synthesis should be clear enough, describing question, search strings, methods, sources of evidence, communicate complexities, acknowledge assumptions, very important, acknowledge limitations and uncertainties, because a research synthesis, we will not gather the answer for everything, okay? And should be accessible, okay? Written in an accessible language and available in a suitable time frame, okay? And now we know, and you already know, that there are a lot of repositories and a lot of softwares. Web of Science, Scopus Web of Science, PubMed, Cochrane, Seal, and a lot of softwares too, okay, and packages that could help us in this challenge of synthesizing and mapping evidences. If you merge synergies between data, articles, between softwares, maybe between artists, we can gather useful, transparent, and up-to-date information. And when I'm talking about softwares, I'm talking about R, QGIS, ArcGIS, Voice Viewer to produce beautiful landscapes, beautiful scientific landscapes, Gephi, among others, okay? Today I will present you a brief tutorial in R, okay? I don't present a new software, or a new package, or a new script. I adapted a script to show you how can we make sense and extract some information from data, okay? Now I will show you a small portfolio of some maps I did. For example, this is a global scientific production and international collaboration map about antimicrobial resistance in wildlife. Okay, the figure was recently published in an article, One Health journal. It's an example. This is another one. Here, this figure represents the location of peer-reviewed reports and field studies since 1957 on the paper, and in the paper that is in evaluation yet, antimicrobial resistance in food-producing animals, a global review. And in addition of offering a broad view of the existing research trends, this study allows to identify research gaps, both in terms of geographical incidence. You can see, for instance, that there are areas overrepresented and areas with not so representation, and of course in relation to unexplored subtopics. Okay, other example, geographic distributions of number per specific livestock species, okay, and you can see that antimicrobial resistance research in food producing animals in low and middle income countries is markedly growing, okay, reflecting changes in food animals production systems, but also, of course, posing some significant threat, not only due to intensive animal production, but also could be exacerbated by poor sanitation and bioremediation infrastructures. Okay, so these maps allow us to sum up the evidences and allow us to produce clear discussions about a specific issue.
João, sorry. These maps were created with R or other software?
R and ArcGIS.
Okay, thank you.
But we can do it with R. However, we have to spend some time trying to code, to editing the code, so it's a bit more difficult. But we can do that. Okay.
Okay, thank you.
But here, okay, we used R to extract all the information. Okay, and then I used QGIS or ArcGIS to map, to project the information, because all baseline information was extracted with R, and I will show you how. Okay. Okay, distributions here is distribution of publications by country and by bacterial genus in another review. Okay. This one is a nice study we are doing now. Here we can see the Earth surface covered by terrestrial biomes of the planet, and dots represent the location of published research, and black colors refers to livestock, green colors wild ungulates, and the proportion of large herbivore species with respect to the large herbivore community has also been represented by the green P and white P charts. Okay, this is a work entitled Incorporating Large Herbivores in Biogeochemical Cycles, a systematic review across biomes. Okay, so again, we have a question, we extract all the information available for the data sources, we gather the information we want, and we map the information using software you want. Okay, and you have other examples, a simple map showing the limitation of some study areas in Pyrenees here, just to see that we can use map for different purposes. Here, this one represents a limitation of an area proposed for classification. Okay, we have an area identified as a good area for eventually represented a new protected area, and you did all baseline information, inventory, etc., and promote and expand this information and represent all this information as a map. Okay. Other one, a spatial gradient of environment favorability for a species, for an ungulate species. Field data coupled with environmental favorability information allow us to identify areas that could serve as potential reintroduction or restocking, but also to identify different areas for habitat recovery, restoration. So maps today could be used, are transversal, could be used in a widely and several research fields, and are really, really useful. And maps could be dynamic too. Okay, for instance, this is a map using crowdsourced data collected over the next 10 years, and here is projected because each information has a date, and this is projected in the map, and we can present this information to the authorities, to policymakers, and they gather information about dispersion dynamics, where we can expect that species will be in the next 2 years, 3 years. Okay, so maps, dynamic maps, static maps could be useful in that way. Okay, here we used validated crowdsourced data on precise location of 50,000 records. So it was.
João.
Yes.
Just one question. If you want to just publish, imagine a kind of dynamic map in a paper or something similar, you can put the map as it is, or you have to put like a link to a dynamic map?
Well, you have to put as a link, as a supplementary information, or even there are some journals that can allow to put it as a graphical abstract, for instance. Okay, you can use as a graphical abstract. But interesting could be, as everything is online, everything is PDFs that allow to put the video, insert the video, okay, incorporate the video. But I don't know any journal that did that right now. But as graphical abstract, you can include it, and you can include it too as supplementary material. Okay.
Okay. Thank you. Yeah.
Okay, so this is some examples, and now I suggest to hands-on. Okay, let's try an example. And I never did research on polar ecosystems. I just bring this example because I guess it is more interesting for you because you like it, polar research. And we already know, and we all know that polar ecosystems are one of the most sensitive environments to the climate change. And therefore I used Web of Science, just an example, okay? And I searched for articles published in English over the last 20 years, from 2020, and searching for these research topics, okay? So we opened Web of Science using an advanced search. I used these search topics. It means TS, Arctic or Antarctic, TS, biodiversity and climate change and global change. This symbol means that we consider climate change or climate changes, global change or global changes, okay? This is like a wild card. And I did my search using these two operators, these two strings. And software, what he did, he goes to the articles, to published articles, and search for these elements in the title or in the abstract or in the keywords. I exclude the main text because otherwise the number will be terrible, because everything he found article in main text, it could be a reference, it could be just an example, and the paper is included in the analysis. So I exclude this search, and I just use the abstract, the title, and the keywords. So every article published in English over the last 20 years that has one of these combinations in the title, abstract, or keywords will appear, okay? And of course, it must be stored in the Web of Science repository. And this is why, most of all, we used Web of Science, Scopus, and other platforms, because there are examples that there are articles that are stored in the Web of Science but not in Scopus. Here, in this case, just for proposal simplification, I just used the Web of Science. Okay. And now maybe we can perform. Let's see what you have here. Okay, so just see, we have here the Web of Science. We use the strings. We have two selected pages. The results were, we gather 111 results here, okay? And we have to a marked list, and then we download it. Here in the marked list, we have 3 steps, okay? We can select everything, every information about the article. We select the authors, the abstract, the addresses, the number, the title, the number of cited references, the source, type of document, keywords, everything. I download everything, and I download it using not TXT, but BibTeX. Okay, I use this kind of extension, but obviously I could use it other extension too. Yes, and then we'll use now R. We will adapt a script from bibliometrics, and then we can project all the information in ArcGIS, for instance, or for guys that for non-coders or hate coding, there's no problem. Okay, we have the guys that develop this package also develop open source and graphical user interface called BiblioShiny that is very, very simple to use, and it's amazing, and allows us to download images in Excel format or TXT format. So if you don't like it, the maps or the graphs they present us, we can download the raw data and develop our graphs and share this information with artists to develop other kind of of representations. Okay, so my suggestion now is for those that installed RStudio, please open RStudio and let's try this example. Okay, if you have any question, please ask me. If I know, I answer. If not, I don't. Okay. Very well. So I will interrupt my share screen and I will open RStudio. I don't know if you are familiar with our language. It's not difficult, but
I think we have a lot of different backgrounds here, so I think we'll have experts almost and not familiarized at all.
Okay. I will do. How many time I have? Sorry, José Guilherme.
45 minutes, I believe.
Still 45 minutes.
Yes, yes.
Okay. So we have enough time, and I will show you briefly how RStudio works. Okay, and then we will open script I shared with you, and we will run the script step by step to see the results. Okay, so I will share. Can you see it?
Yes, yes.
RStudio? Okay, perfect. So we query WOS, Web of Science. We downloaded the information, okay? The information was stored in a BibTeX that I shared with you later and call it Polar Research WOS, I guess. And now we will use this information. This information will be opened in RStudio, this graphical interface, to be treated, to be extracted information we want to analyze. So RStudio, this interface we show here, is a graphical interface where we can write, edit, and save our code. The interface, okay, is divided in 4 windows. This is just 3 because I have to create a new file that is our coding window. Here we will add, we will write, edit, and save our code, okay? The upper left corner here is where we write our code. The lower right corner, the lower left corner, sorry, is where it is presented numeric and tabular results. The upper right corner lists our data sets, vectors, variables, lists, and so on, maps, shapefiles, okay? The lower right corner, we can find packages available, and we can find the plots, okay? So just to see, I can create a vector. This is a vector. This is basic elements of R, a list of name, for instance, of numbers. I can create Guilherme, José, let's see, Carmen, Carolina, Joana, Carmen. So I can create a vector, Carolina, Joana, okay? So let's see, okay. So I create a vector with 5 names, okay? So I can perform for in this, for each element in the vector V, please did that, okay? Print past, for instance, something like that, so each element, virgula, welcome to art and science workshop. So, okay, this is a vector, this is a kind of loop. Let's see. So our code, our results, our data frames. If you want, here we have our packages and our plots. For instance, we can create a plot. Okay, it's just to see. Yes. Okay. It's an X, and we have our plot. So this is our 4 windows. Here, where we write our code. Here we have the numeric results. Here we have the data frames listed data frames, and here we have the graphic results. And when we open now the script we send it to you, we will run every lines, and we will see this kind of information appearing in each section. Okay, so let's do it. File, open file, and then we can search for the script. There is here data and polar research. No, this is file. This is not script. This is file I downloaded from the Web of Science. So as you see, it's a file with a lot of elements. It seems not so well organized. I want to turn this file in a data frame divided where each column represents something. I will have a column for authors. I will have a column for title. I will have a column for journal, year, volume, month, and so on. Okay, and so this information was downloaded from Web of Science, from our Web of Science. This is a paper, and then we will have other paper, and we'll have a lot of papers, a lot of lines. Okay, then we want to translate this in an editable and friendly data frame. Let's open file. Let's see where is the code. And this one. Okay. Yes. So I will delete all the other things. If I want to delete this console, Control L, and it deletes. I can drop this. Oops, I can. Don't save. And okay, this is what we got. Okay, this is nothing new. I adapted this tutorial from Massi Maria and Corrado Cucurlo that is available in this link. And this art is so beautiful and so interesting because there's a lot of available information in the internet that you can use to answer our purposes. Okay, so the first thing we must do is to set our working directory. Okay, so where R is working? Okay, R is working in the C users J documents. But now here in the set working directory, we must say to R that from now on, we have to read all the elements in this folder, in this specific folder. In my case, is named C users. I saved the file, the information file with the references in this folder. But you, please, you cannot use this folder. You have to copy the link where you stored the bib file and paste your link right here. Okay. Please note that you have to use 2 backslashes or 1 /. Okay, if you only used 1 backslash, the R don't recognize the path. Let's see. So get, okay, 1 backslash, R don't recognize it. So to define your working directory, your path, the area where you, the folder where you save the file, the BibTeX file, you should set working directory with 2 backslashes. And now I guess there's no problem. And okay, now I have our working directory defined. If I want list files, I just use list files, and R will list files that are stored in this folder. And in this list of files, the file we want is that one, polarresearch.bib, bibtext. This is the file we downloaded from the Web of Science, and this is the file that contains all information about each article. And this is file that will edit and work, okay? But before that, we have to install packages, because R, as a software, a light software that comes with several functions, but has more or less 70,000 packages. And bibliometrics is just one of these 70,000 packages. And to use these functions, we must install packages. We must install bibliometrics. This bibliometrics package provides a set of tools of quantitative research in bibliometrics, okay? This quantitative evaluation of publication and citation data includes, for instance, and allow us to evaluate the growth of a scientific field, maturity of a scientific field, recognize the leading authors, the conceptual and intellectual maps, allows to perform conceptual and intellectual maps, trends of scientific community, and so on. A lot of information. So the next step, please, if you have any doubt, please ask me. Next step is install bibliometrics. Okay, so it's installing. Okay, but now it's installed. But now I have to say to R, please R, okay, I already installed software, but now I need that all functions are made available, and therefore we must use the function library, and I will library. Okay, it's processing. Okay, when you found a stop here, is that software is processing. While software is processing, please do not click in other buttons, in other options, because it may crash. So every functions are now ready, available to be used. But for instance, if you are using this package for your thesis, for an article, for a report, and you don't know how to mention, how to set this package, you can use this function, okay? And this is R, is amazing, and is a good friend of us, because with just this argument, they give us the reference, okay? Just copy, paste in your article, in your thesis, okay? Very, very straightforward. Okay, so the first step is to convert BibTeX, the file that with a lot of information, but nothing friendly, a lot of information that is not be so organized. So I want to convert this BibTeX to a data frame that can be read, edit, and worked in R. So there's a function that is from bibliometrics, that is available in bibliometrics, that allow us to convert different data sources from Easy, Web of Knowledge, data sources data sources from Scopus, data source from other sources, okay? Different formats, in this case BibTeX, will convert in data frame, okay? Imagine you have two files. In this case, we only have one file. If you have two files, we just need to C, concatenate, and name the files, give the list of files, and it will be open all these files in one data frame, okay? So let's try it. Okay, it's reading, it's converting, and awesome. It generates a data frame called D with all information available. Now, in 2 seconds, our software converted this file was converted in this file. Authors, keywords, abstract. There's a lot of information. DOIs, digital information, countries, journal, year, and a lot of information here. Okay, so this information was converted in a data frame, and you can see data frame. Now the first step is obviously to sum up the data frame. Wait, we have a comment here. I can see the comments. I guess we have the comment, Guilherme.
Yes, so the person is basically saying that he has an error in the working directory, that he cannot change it.
So please go to the folder where we stored the file, the polar research WOS.pib, and just copy the directory. After copying the directory, paste the directory here. Don't forget the symbols, don't forget the backslashes, and read. Okay, here we just need to name the folder. We don't need to have the name of file, the name of the bib file. Okay, just name of folder. If we are not able to that, at the end of the session, I could in private solving this issue. Okay, don't worry. But using this, I guess you will have no problems. Okay, just try again, please. So the so we export file, read and convert to data frame using the argument db source and format bibtext. Now the idea, and because each manuscript has several elements, authors' names, title, keywords, and so on, the first step is to perform a descriptive analysis of the bibliographic data frame. The function biblioanalysis, as we have here, calculates the main bibliographic measures using this function. Okay, this is really simple. So we can report separated by semicolon, data frame. Okay, data frame is separated each column by semicolon, and let's do it. And okay, so now we have here a lot of information. We have here the words, the keywords. There are so many that even though the information is hidden. Okay, but no worries, because now we can limit the number of keywords, the number of journals, the number of countries we want to represent, to minimize the information represented, and then it allows us to confirm the main authors, the main countries, the main subjects, the main journals. Okay, so using this report, we limited number of for instance, number of countries we limit to 10. Okay, just 10 the 10 most productive countries will be presented. If you want most 20, we just have to change to 20. If you want 5, we just change to 5. But let's see. So, can you see it? What we have here? We have a summary. Time span from 2000 to 2020. Sources, 113 sources. So, between journals, etc., we have 113 different journals. Documents, we have a total of 189 documents. Average years from publication, average citations per year, per document, etc. Document types, we filtered for articles, but sometimes we downloaded other documents too, and we can of course subset this information because if you are only interested in articles. Okay, but otherwise we will continue in this example with everything, but mainly we have articles, we have 2 book chapters, 6 early access, okay, papers that were published. Yes?
There was a question. The result says not found. What can I do?
The results not found here. Oh, here.
I
In
believe.
summary.
I think so. Maybe the participant can.
Hello.
Hello.
Yeah. Well, I was doing all the things you were saying,
Yeah.
and in the console, in the main console, says that I don't have. I have an error in summary object,
Here.
and it says object report not found in with S summary.
Okay. But you run this line of code?
Yes.
You run it? Okay. And you have the data file here listed? You have the information here? You have a data
No,
frame?
I don't have that.
So it's, if you don't have it, have the data frame, so the error is, your error is here, okay? You have ensured that you are able to read the data frame, okay? Please, maybe in the set working directory, maybe it could be a error here, but sorry, just not to be too late, because we only have 20 minutes. I suggest before the end of the session, I could combine something 10 minutes, 20 minutes after that, and I'll try to help you.
Okay. Yeah.
Just to do everything I have here. Sorry, okay? But then I will try to help you.
Okay.
So
Thank you.
authors collaboration, annual scientific production, okay, the number of papers by here, the annual percentage growth rate, most productive authors, top manuscripts per citation, corresponding authors, etc. Okay, we have a lot of information, the keywords, and so on. We can, well, we may want to represent only the top five. And therefore, this argument allows to represent only top five. Okay. Reports, simple functions could allow us to translate in tables. These tables can be then can be exported and analyzed. Okay, global change biology, the number of papers per journal, country collaboration. We have USA single author papers 20, multiple country papers 15. This is you can see who is more friendly or not, more collaborative or not. We can also gather some plots. Well, the arguments are too simple. Let's see. Okay. Some plots here are not really beautiful, but well, these plots were performed with the ggplot tool. But all this information is we can be downloaded and perform and produce our own plots. Okay, our own information, because these plots are not really too beautiful. Okay, I can hit. Okay, using the pause through, I have to hit the console to show one graph at a time. So you can see, using a stream, Web of Science, Scopus, we can gather a lot of information. Of course, if you want to have more detailed information about where the study were performed, what are the main results, to a meta-analysis, for instance, we have to read one by one. And the graphs I show you earlier, we have to read one by one to show reference all the field studies in the different area. Okay, but this kind of information, most productive authors. Okay, well, this is an error because we have to consider just complete cases. We have some null values, therefore I will consider just completed cases. We see completed. Okay, and here I have a top authors production. Okay, there are guys that publish more than one paper in this subject by here, the number of citations by here. Okay, so it's a kind of summarize about this kind of sum about the maturity and interest of this area. Okay. Bibliographic coupling. Bibliographic coupling represents the, for instance, two articles are said to be bibliographical coupled if at least one cited source appears in the other bibliographic or other paper. For instance, articles that share only a few references, therefore will tend to be weak, clear, bibliographical couple. The papers that share a lot of references, well, in terms of bibliographic couple is too many, and perhaps the articles are much closer than to others. Okay. And we can produce scientific collaboration by country. Let's see. Okay, authors coupling. So we have a network here, an example of network is thinking. Just okay, you have an authors coupling. The authors are linked to other authors. When the link is strong, is that number of collaborations is high. When the link is weak, is that number of collaborations is lower. Okay. And this is other example about let's see, where is the art plot? Let's see, where is Zoom? No. And we can export information. This is a network that represents the collaboration, the country collaboration. Okay, we have a USA with a lot of articles that is linked to many other countries, representing a collaboration network. Now I try to zoom in but I don't know, I can't. I'm trying to zoom in the image, but now it doesn't appear. But believe me, this is a network. When I zoom the image, we can see the links between countries representing the number of collaborations, okay? Co-occurrences of keywords. And this is a nice, and once again, well, I'll try to save the image to show you. Okay, I can save the image, for instance. Okay. Okay, here it is, keyword occurrences. Can you see it?
Yes.
Okay, great. So we have here some clusters to identify, for instance, subjects and interesting areas of interest. For instance, climate change is too linked to responses, environmental change, impacts, Arctic tundra, ecosystems, global change. Species richness are linked to biodiversity, patterns, evolution, biogeography. Temperature is linked to climate, marine variability, ecology. So it could be useful. And this network is not properly beautiful. It's not really beautiful, but the advantage is that we can export this information to other softwares, Gephi, Pajek, even Excel. Excel has a plugin called Node.xl that allows us to perform beautiful, beautiful keywords. And even in R, there are packages specialized and developed properly and specifically to map these keywords of co-occurrences, social networks, geographic networks, iGraph, for instance. There are a lot of packages that allow to perform this kind of analysis. And of course, there is co-word analysis, for instance, some dendrograms that allows to map the conceptual structure and so on. Obviously, these graphs are not beautiful, but my idea is just to teach you how we can extract information. Then you extract information and the number of hypotheses, it's a kind of imagination. We can represent this kind of information in very different ways, okay? Obviously, these graphs are not most beautiful, but okay. So this is a simple code to extract information from our databases. But as I said before, the authors also developed a small application, a user-friendly application that can you use it? We can use it. It's called BiblioShiny. So everything we have done here using code, we can do it using just a few clicks with your mouse. So BiblioShiny for bibliometrics, okay? First step is to import our load files, of course, okay? Import raw files, database, Web of Science, but could be Scopus, I mentioned, PubMed, Cochrane. For instance, imagine when you are using Web of Science, we just downloaded 500 articles at a time. But if you want to download 1,000, 2,000 articles, for instance, 2,000 articles represent 4 big text files. And this particular application just can read 1 file at a time. So it can read only 500 references. But if you previously edited, you can edit these 4 files and merge all these 4 files in just 1 file using Mendeley, for instance, and then export this information and read here in BiblioShiny, and then we can read 2,000, 3,000 articles at once, okay? Otherwise, we are able to read just 500 at a time, because when you download information from Web of Science, it's just allowed to download 500 articles at maximum per document. Import raw files, Web of Science, plain text, no bib text, and choose a file. Let's choose a file. Polar Research, is that one? I guess so. Okay, is that one? And start. So easy, huh? I guess that for master thesis, PhDs, for an introductory chapter, for instance, it could be useful, this kind of analysis. You have authors, you have keywords, and this, if you take a look at the website of Bibliometrics, and this is encoding and the meaning of each acronym, it's available, authors, keywords, and everything else. We have a lot of information, and now I can extract information very, very easily. Annual scientific production. Oh, is here. The graph. I don't like the graph. It's ugly. I don't like it. No problem. We have here a table that we can download it in CSV, Excel, and then open in R, MATLAB, Excel, SPSS, Origin, and doing our own graphs. Okay. The average citations per year. No, okay. Oh, so ugly, but no problem. We can download it. Okay, the information. Sources, most relevant sources, most local cited sources, source impacts. Author, most relevant authors, author impact, most cited countries. Okay, apply. Most cited countries, we can download it. Average citation countries. Documents, most word cloud. We can perform a word cloud. Biodiversity, diversity, okay? We can change the parameters, frequency, square root, log, font type. We can change it, okay? It's beautiful. Background, color, rotate, and so on. Keyword plus, author keywords, titles. Let's see abstract, for instance. The abstract, the sentence, the words are different, okay? So we have a lot, tree map, okay? Climate change is the most used term, most used word. Word dynamics, and this is awesome. Word dynamics. Over time, how is evolved the interest of researchers by the field of climate change and biodiversity in polar ecosystems. So climate change is in exponential growth, but there are others, Arctic tundra communities, that over the last years, the interest is decreasing, okay? There are others, responses, diversity, that are increasing slowly. Climate is increasing slowly, climate change too much. Well, this is raw data. Perhaps you must use, and you must filter some terms, okay, and some words. But this is background work that you must do before. Biodiversity, it's increasing at same rate of climate change, but then it seems that decreases. So I guess it's a nice map to see trends and where we are and where we expect to be in the future. Co-occurrence network. Okay, the networks are here. Imagine, I don't like it, this graph, no worry. We can save as figure or PAGEC. PAGEC is a file format that could be used to open in other files, in other softwares. Gephi, for instance, PAGEC by itself, or Voice Viewer, and you can, Voice Viewer is nice. Voice Viewer software is very easy to use and allows to perform some scientific landscapes, okay? It's very, very useful and very easy to use. And for instance, conceptual thematic map.
Sorry, João?
Yes, I will see.
We have just here a question that BiblioShine can be only used for Biblio data?
Yes. Yes, I will show you the kind of files they read, but yes, okay. I will show you the kind of files, but it was developed for bibliographic data, okay. For instance, this country collaboration map, it's not particularly beautiful, so we can export this table, the collaboration between Argentina and Brazil. And APECS, for instance, I saw in the objectives of APECS is to promote collaboration and promote, so we can see if you are really collaborating. So Argentina, Brazil, 2 cases of collaboration. Argentina, Chile, Portugal, where is Portugal? Let's see Portugal. Portugal is here, Portugal, Italy, Russia, Portugal, Spain, Portugal. So we can have an idea about collaboration, we can export this information, we can associate to this information a centroid of the country and create maps like I show you in the presentation, okay? So this is a way of sum up evidences. Of course, this is raw data. It is not solution for everything. If you want to perform a meta-analysis, a systematic review, you have to read each paper, okay? Abstract, results, study area, to be able to identify the area where the study were carried out and so on, okay? But just to show you that there are a lot of hypotheses, a lot of software, a lot of synergies that can be promoted to make sense about data, that make sense about information, that can sum up the evidences from a wide number of thousands of papers, okay? That now we are living in a information era and we have to take advantage of this kind of approaches, this kind of software, using our imagination to make this information accessible and useful for decision, for decision making, for decision policy makers, etc. Okay, so we can use it, this kind of software. I will show you the presentation. I can share the presentation with you, some tutorials too, and okay, and this is it. Because it's nice, because life and biology and ecology and everything, nature, okay, is driven by simple rules, but of course by complex relationships, and therefore we must use synergies and we must promote synergies to connecting the dots. Art and science are just one of really important synergy to make sense about data, to interpret and to represent our data. Well, it was 1 hour and a half, more or less. I hope you enjoyed. I hope it is useful. It was very, very small tutorial, but okay. I hope I can show you some options to explore, and it could be also useful for your thesis, for your research. I don't know. I hope. Thank you. Okay, I finished.
So thank you, João. I think if anyone have a question or want to put a question to João, is free to ask. Or
just one thing. Sorry. Okay,
yeah.
Jose.
Go.
I forget. As said Jose at the beginning, I am the coordinator of this publication, Lucanos, Revista de Ambiente e Sociedade, that tomorrow we will release our fourth volume. That the aims is very clear, is to get close academia, science and society to answer specific problems, and maybe we can take a look at this site to see the previous numbers, and maybe tomorrow or next week the fourth volume will be available too. And we try to communicate science also through some art, some nice graphs, and you are all invited to submit your papers, your contributions, because the idea is to translate our knowledge, what we are doing in the academia, to society, okay, in a beautiful I guess in a beautiful way. Okay, so thank you.
Thank you, João.
Yeah, thank you for doing this presentation also in a really tight schedule as you have that important thing tomorrow. Thank you so much, it was amazing. So does anyone ask questions for João? It's your last chance. Okay, we have one. Is there similar softwares, for example, species representation in world maps and so on, with genomes databases, for instance?
I guess so. To represent this kind of information, I use ArcGIS, QGIS, or R. But so nice, so user friendly, I didn't know for this kind of representation. But ArcGIS or R could serve your purposes and meet this objective or representing.
I think
this is representation in world maps. Well, I used ArcGIS or QGIS or R, are the main. For those participants that have doubts during the session, I could be here or in other, I don't know if it is possible, but I can leave my contact. I will leave my contact here and they can ask me, okay?
Yeah. Yes, and also the session will be available to consult later if you want, to all the participants. So I think it's more easily so to go through the presentation then.
Yes, and feel free to contact me whenever you can and whenever you want. Okay? Thank you, Guilherme, José e demais colegas.
Thank you so much. I think we only have one more thing to do, which is to try to take the picture of all of us, to take like this memory. So maybe those that don't mind to, turn your cameras on, they could do it. Those that don't want is also fine, no worries. Okay.
Just for a symbolic one, so
yeah, just a memory, so no worries.